Hi, today we will talk about nucleic acids and we'll take a look at the structure of DNA and RNA. As carbohydrates and amino acids, nucleic acids are considered macromolecules, meaning that they consist of building blocks called monomers, which in our case are nucleotides. Let's take a look at the structure of a nucleotide. A nucleotide is composed of three parts: a phosphate group, a pentose, so 5-carbon sugar in ring form, and a nitrogenous base. The sugar in RNA is ribose, and in DNA it is a deoxyribose. The only difference between these two sugars is that ribose sugar has a hydroxyl group at position 2, whereas deoxyribose sugar has a hydrogen atom at position 2. The way we number carbon atoms in sugar molecules is the same as in other organic molecules. So, if we take the linear representation of the ribose molecule, we will see that this carbon is a part of a functional group, which is the carbonyl group in sugars. So this will be our first carbon. This also translates to the ring form of ribose. So the base is attached to the 1' carbon, and the phosphate group is attached to the 5' carbon of our 5-carbon sugar. Now let's take a look at the nitrogenous bases of these nucleotides. We can divide them into two groups, purines and pyrimidines. Purines in DNA as well as in RNA are adenine and guanine. Pyrimidines, however, differ. In DNA we have cytosine and thymine, but in RNA instead of thymine we have uracil. You probably noticed that the difference between these two types of nitrogenous bases is that purines have a 2-ring structure, while pyrimidines only have 1 ring. This characteristic will affect the way they are connected to each other inside of DNA. Let's begin with the structure of DNA. You probably have seen illustrations like this all the time, but now we want to dive deeper and see the actual structure. A nucleic acid, as well as an amino acid or a carbohydrate, is a chain of monomers that are connected by covalent bonds during the dehydration reaction. In the case of nucleic acids, such as for example DNA, the phosphate group of the 5' carbon attaches to the 3' carbon of another nucleotide, in the process called the DNA replication. When this process is completed, we get a chain of nucleotides that begins with the 5' end of the first nucleotide and ends with the 3' end of the last nucleotide. But we all know that the DNA molecule is double-stranded, so how does the other strand look like? Well, turns out that the most stable way for two strands of DNA to be connected is for them to be anti-parallel. This means that both strands are oriented in the opposite directions. What we can also say about the structure of DNA is that its strands have sequences of bases that are complementary to each other, meaning that every pair of bases connected by hydrogen bonds will always follow the complementary base pairing rules. Adenine always pairs with thymine through 2 hydrogen bonds, and guanine always pairs with cytosine through 3 hydrogen bonds. And so that is how the well-known double helix structure of DNA is formed. But what about the RNA? Most of the times we hear about DNA, and of course it is the main structure storing our genetic information. However, this was not always the case, according to the RNA world hypothesis. The idea is that in the earliest organisms on our planet, RNA was the molecule storing and transmitting genetic information. Although now it's not the case anymore, RNA still plays a major role in various cellular processes, including protein synthesis. The structure of RNA is similar to that of DNA, with differences in nucleotides that I mentioned earlier. But the main difference in the composition of the nucleic acid itself is that RNA exists as a single-stranded molecule. This allows a single strand of RNA to fold into complex structures, similar to the way proteins do it. tRNA, the molecule involved into protein synthesis, is an example for this. Thanks for watching, and I'll see you in the next one.